Okay, let's unpack this. Today, we are diving deep into what might be the most chaotic and, frankly, most successful driver of aggressive cancer, extrachromosomal DNA or ecDNA.
Yeah, and when we talk about xDNA, you have to remember these structures are, well, they're fundamentally different from the rest of your genome.
They're not normal chromosomes.
Not at all. They are these huge megabase-sized circular DNA episomes that just float freely in the nucleus, completely separate from the main chromosomes. They're essentially genetic renegades.
And the problem they present is immense. I mean, they're major drivers of malignancy because they allow for this unchecked oncogene amplification.
And they accelerate tumor evolution at a frightening pace.
Right, and they generate this massive intratumoral genetic heterogeneity, meaning, every single cancer cell can be slightly different from its neighbor.
That instability is precisely why they are so dangerous.
Yeah.
We know that xDNA-derived amplifications are detected in about, 17% of adult tumors?
And that frequency just climbs. It goes way higher when you look specifically at metastatic, hard-to-treat tumors.
Absolutely. This kind of instability is a fundamental marker of poor survival.
So ecDNA isn't just adding copies of a bad instruction manual. It's a major form of genome instability. But the core question we're tackling in this deep dive is how these unstable circular structures act as major platforms for generating highly potent oncogene fusion transcripts.
And that's the key. We're not talking about just copying existing genes. We're talking about generating brand new, hyper-effective genes by splicing different parts together.
Think of it this way. If a healthy genome is a regulated factory with clear quality control,
ecDNA is a chaotic, high-speed assembly line. It cuts up blueprints and welds the most aggressive parts together. It's a process of rapid, chaotic, I'd call it chaos engineering.
to produce these optimized super instructions for cancer cell survival.
Exactly. So let's get into the evidence that reveals this chaotic landscape.
All right, before we get into the data, we need a quick definitional check. The underlying genomic damage that creates these new instructions, it all starts with structural variants, or SVs.
Right, that's the rearrangement itself.
Yeah.
The translocation, the inversion, the insertion that abnormally joins gene segments that should never touch.
And the functional result of that SV is the gene fusion, or the fusion transcript.
That's the products. It's where two genes are merged, leading to altered regulation or sometimes a completely novel protein function.
Yeah.
What's amazing about this research is the sheer scale they use to map where these events happen.
It's huge. Researchers integrated whole genome sequencing data, and they use a specialized tool called Amplicon Architect, which is really designed to identify these circular ecDNA structures, and they combine that with RNA sequencing data from over 1,800 tumor and cancer cell line samples. This wasn't a small survey. It covered major databases like TCGA and CCLE.
And the conclusion was, well, it was inescapable. ecDNA is the definitive fusion hotspot in the cancer cell.
A hotspot.
They looked at the rate of structural variants supporting gene fusions. They call it the SVGF rate. And xDNA had the highest frequency by far.
And that was compared to everything else.
Everything. All other forms of copy number alterations, including breakage fusion bridge structures or just standard linear amplifications, ecDNA was number one.
So it's not just unstable, it's selectively unstable in a way that generates these highly functional fusion transcripts. Is the connection between the SV and the resulting RNA fusion really that tight?
Oh, it's remarkably tight. The data showed that 88.5% of the RNA fusions they detected on sDNA were directly supported by an SV they could pinpoint.
Wow.
It confirms the circle structure itself is actively mediating the rearrangement that creates the final product.
And just look at the prevalence. While only about 14.5% of the total cancers they analyzed had these ecDNA-borne RNA fusions.
That number just exploded to over 55% of cancers that were already known to be xDNA positive.
So if you have xDNA, you have a better than even chance of having these potent fusion transcripts.
That's right. And they aren't benign. These fusion transcripts that come from xDNA showed significantly higher expression levels than fusions happening anywhere else in the genome.
And the highest output, that was consistently found when the cell had both the RNA fusion and the ecDNA amplification.
Exactly. The circular nature allows the gene to be copied at a higher rate, which turbocharges the output of the new fused transcript.
So we have hyper-unstable DNA leading to hyper-expressed transcripts. But what really stood out to me was the tissue specificity. This isn't some generalized attack, it's a tailored one.
And that's a crucial finding with real diagnostic implications. For example, the sources noted that PVT1 and MYC fusions were highly enriched in lung cancer.
Right.
ERBB2 fusions were prominent in breast and upper GI cancers. Meanwhile, The aggressive CNS tumors, like glioblastoma, were dominated by EGFR fusions.
And MDM2 was the key player in soft tissue sarcomas.
It's like ecDNA is leveraging the specific genetic vulnerabilities of different tissue types. It's incredibly clever in a sinister way.
Okay, let's take a quick moment here to recap that first chaotic landscape. ecDNA is ground zero. It harbors the highest rate of structural variants, which directly supports the highest rate of these highly expressed RNA fusions.
And these fusions are tissue-specific. This really paints ecDNA as the ultimate genomic engineer for cancer fitness.
Which leads us straight into the core question. Given this platform of chaos, why does one specific long non-coding RNA, PVT1, emerge as the most frequent fusion partner?
It has to be doing something more than just being amplified. What is its secret weapon?
So PVT1 is definitely the dominant player here. It was the most frequent fusion hotspot in xDNA-positive cancers, showing a stunning, a 4.4-fold enrichment compared to fusions found elsewhere.
And it generated the greatest diversity of fusion transcripts. Almost 3/4 of all PVT1 fusions, they arise directly from xDNA.
And the structural pattern is remarkably conserved, which that suggests strong evolutionary selection.
It does. Across the board, 98.5% of these PVT1 fusion transcripts have PVT1 positioned right at their 5' end. And the fusion break always happens very early, typically in PVT1 exon 1 or intron 1.
Let's hone in on that exon 1 piece for a second. It is tiny, only 7 to 12% of the native long non-coding RNA.
Yet it's retained in 95% of the amplified fusion transcripts.
That's incredible. The fact that the cancer cell selects for this small segment 8 to 14 times more often than you'd expect, it tells us it confers a massive survival advantage.
And the quintessential example is the PVT1 MYC fusion, the most prevalent one. When it forms, it literally swaps out the canonical MYC promoter and exon 1 for the PVT1 promoter and exon 1.
So the researchers had to figure out, is this fusion just hyper-expressed because there are more DNA copies, or is something deeper happening post-transcriptionally?
And here's where the experiment was just brilliant. They used isogenic cell lines. The COLO320DM line, where the fusion lives on the scDNA,
right,
and the COLO320HSR line, where the MYC amplification is actually integrated into a standard chromosome.
So by using these two lines, they could compare apples to apples, normalizing for the total DNA copy number.
And the finding was completely counterintuitive. Even after they mathematically corrected for having the same number of gene copies, the steady-state PVT1 MYC mRNA level was still 2 to 3-fold higher than the canonical MYC mRNA.
Wow. So that finding was a clear signal. Amplification isn't the whole story.
Not at all. Something is protecting that RNA after it has been made.
The PVT1 tag acts like a preservation system. So how did they confirm that stability mechanism?
They used a classic RNA stability assay. They shut down all new transcription using a drug, actinomycin D, and then just watched how quickly the RNA decayed.
Measuring the half-life.
Exactly, the decay rate, beta. And consistently, PVT1 fusion transcripts decayed significantly slower across multiple partner genes and cell lines. That PVT1 segment provides superior RNA stability.
Okay, so that brings us to the cell's natural disposal system. Canonical MYC mRNA is meant to be short-lived. The cell has checks and balances to make sure it doesn't overproduce.
Right, and the primary check is usually a mechanism called nonsense-mediated decay, or NMD.
Nonsense-mediated decay, that sounds intense. What is the nonsense it's detecting?
Think of NMD as the cellular quality control police. It actively degrades mRNA molecules that look defective, and it's often triggered by the translation process itself. If you block translation with a drug like cyclohexamide...
The NMD police get confused.
Right. And the canonical MYC mRNA stability actually increases because its main decay pathway is suppressed.
Okay, so blocking translation normally stabilizes MYC. What happens when you try to block translation in a cell that has the PVT1 fusion?
And that's the smoking gun that confirms evasion. Blocking translation did stabilize the canonical MYC, as expected, but it did not further stabilize the PVT1 MYC fusion.
Which means?
It means the PVT1 exon 1 fusion enables the oncogenic transcript to completely bypass or escape those normal translation-coupled decay mechanisms. The protective tag shields the instruction manual from the quality control police entirely.
So let's recap section 2. PVT1 exon 1 is the most common fusion element because it confers superior RNA stability. And it does this by helping the oncogene evade NMD, the cell's own sophisticated translation-coupled degradation system.
Which naturally leads to the next question. If PVT1 exon 1 is the stabilizing element, what protein is actually binding to it? What's acting as the protective shield? It has to be a physical interaction.
To find that physical shield, researchers had to perform some real molecular detective work. They used a technique called CRPMS,
Right, comprehensive identification of RNA-binding proteins by mass spectrometry.
which essentially lets them isolate the PVT1-MYC transcript and then identify all the sticky proteins that are bound to it.
And that mass spec approach, it clustered a whole bunch of RNA-binding proteins involved in stability and translation. But one candidate really stood out when they cross-referenced it with structural data, SRSF1.
Serine arginine-rich splicing factor 1. So they needed to validate that SRSF1 was the right suspect.
Absolutely. They followed up with RNA immunoprecipitation, or RNA-IP, which confirmed significantly higher SRSF1 occupancy on the endogenous PVT1-MYC mRNA compared to the native canonical MYC mRNA.
So SRSF1 was preferentially glomming onto the fusion transcript?
Exactly.
And the structural analysis told them exactly where SRSF1 was docking, right?
Precisely. Using advanced computational modeling, specifically AlphaFold 3 prediction, they could visualize the interaction. They found that SRSF1's two RNA recognition motifs interact specifically with the 5' end of PVT1 exon 1.
And it wasn't just anywhere.
No, it clustered near a very distinct sequence motif. It's a very specific binding site.
The ultimate proof of necessity, then, is showing that if you break that binding site, the protection is lost.
And that was the definitive experiment. They introduced targeted mutations into that GAGN motif, and they also deleted the critical 5' 75 nucleotides of PVT1 exon 1.
And the result?
Those mutations completely abolished the RNA stability boost. It immediately reduced the decay rate right back to the canonical MYC level.
And critically, those mutant fusion transcripts immediately regained sensitivity to translation inhibition.
Meaning the NMD evasion was gone. That small 75 nucleotide fragment bound by SRSF1 is the required lock for the decay pathway key.
So it is. This makes the PVT1-MYC fusion a true super MYC. It's not just surviving, it's actually functioning better than the original.
A lot better.
How much better? I mean, we need to quantify that super status.
Okay, so they performed a rescue experiment. They used MYC-addicted cancer cells, the EC4 line, which are completely reliant on MYC for survival.
Right.
When they depleted the canonical MYC, the cells started dying, as you'd expect.
Yeah.
But when they rescued those cells with the PVT1-MYC fusion, it was significantly more efficient at restoring cell viability than canonical MYC alone.
So it's a better survival factor. Does it also act as a more potent transcriptional activator? Does it turn on more downstream genes?
Yes. Using single-cell RNA sequencing, they looked at the core downstream MYC target genes, the ones MYC turns on to drive growth and proliferation.
The hallmark pathways.
Exactly. And they observed that PVT1-MYC expression correlated more strongly with the activation of those target genes in a dose-dependent manner compared to canonical MYC. This fusion transcript is a more effective signal booster.
So, to synthesize this entire journey for our listeners, ecDNA creates a perfect storm for cancer fitness.
It does. It provides the initial amplification boost, that's the high gene dosage, but then, through its structural instability, it performs molecular surgery, swapping in the PVT1 exon 1 element.
And that element recruits SRSF1 to act as a permanent protective shield, preventing the oncogene mRNA from being degraded by NMD.
Resulting in a transcript that is both highly amplified and hyperstable. This molecular engineering enhances the survival and potency of the most aggressive cancers.
So what does this all mean for you, the learner, trying to stay ahead of the curve in clinical cancer research? This deep dive shows that ecDNA isn't just a copy machine for existing flaws.
No, it's an engineering platform. It creates new, highly optimized oncogenic molecules specifically designed for cell fitness and ultimately drug resistance.
The clinical takeaways are highly relevant right now. This strong molecular link ecDNA amplification driving PVT1 fusion RNA expression means that detecting these specific fusions dramatically improves diagnostic accuracy.
Potentially up to 95% when combined with copy number analysis. It gives clinicians a very sensitive biomarker for these aggressive tumors.
And because these fusions are unique to the tumor cells, they're completely absent in normal cells, they represent incredibly precise, clean targets for novel therapeutics.
Things like personalized mRNA vaccine-based therapies. You're attacking a tumor-specific design flaw, not a necessary cellular function.
Which means the therapeutic challenge now really centers on disrupting that stability mechanism. The source material highlights that the 5' 75 nucleotides of PVT1 exon 1, the SRSF1 binding site, is essential for the fusion's potency. It's the ultimate Achilles heel.
It really is.
So if that tiny 75 nucleotide fragment is the key to stability and survival for this entire class of aggressive cancers, What if we could design a small molecule or an antisense oligonucleotide that specifically blocks SRSF1 from binding to only this oncogenic sequence?
If you could inhibit that single specific interaction, you would effectively strip the armor from the cancer cell's core instruction manual. That would trigger the immediate NMD-mediated degradation of the super MYC, essentially forcing the cancer cell to self-destruct, all without impacting normal, healthy gene regulation. That is the therapeutic pathway that this fundamental discovery has opened up.